project, and he was absolutely saying we can do this. He was super excited, and so I very much appreciate him. I very much appreciate the ARC Design Hub and their team and TAC and everyone for working with me on this project. I did have the opportunity to be a professor at ARC. I believe it was for 3 years, and so I got to work with everyone there. And so again, thank you so much for having me here. So what I'm going to talk about today is, well, actually, let me step back and give a little bit more background as to me. So a long time ago, I was a medic in the military. It was something I enjoyed, something I very much like to see how medicine could help people. But I saw that we needed better tools. And I wanted to design better tools for the nurses and doctors, the wonderful people who help and others. And so that led me on the path to eventually get a PhD in computational chemistry at UC Davis. From there, I taught a total of 5 years at community colleges, and then went on to also do an internship at Novartis, and then where I'm at currently, which is Atomwise, which is an AI-powered pharmaceutical company. And I'll be glad to talk more about that and sort of my experiences and any thoughts on that as we go along in the subsequent seminars. But what I want to talk about today is Moleculeze. It's a EduTech project at ARC Design Hub. And I want to talk about how this project is helping the whole process of crowdsourced molecular discovery, medicine discovery, and that's what this talk is going to be about. Okay, so I'm going to talk about these subjects. So the first is, why is it important to find new and better medicines in the first place? Why is that something that we should still be doing? There's this impression that people might have that we basically solved all of the problems and that there are for diseases. And I can tell you that we are actually far from that. And there's a huge need still for us to develop new and better medicines. And then I'm going to describe how cooperation and sharing can actually improve the search for medicines. And also how most medicines typically work, so just to give some background so we can have an understanding when I talk about a therapeutic target or a ligand, what do those things mean, and especially in the context of designing medicine. And then I'm going to describe how education and medicine can be enriched through gamification. And this is something that I'm really excited about, and I know Randy is a big advocate for as well in TAC, where you take a hard problem, like the idea of designing medicine, and you make it so that it is accessible to everyone, and everyone can be a part of it. And you share those results, and that crowdsourced sort of approach makes this finding medicine more efficient and faster. And then I'll share a plan for moving forward and how we can work together in order to make this possible. We'll also learn in subsequent meetings, learn about and practice some tools and concepts that are important in drug discovery. And then finally, I'll discuss my experiences working at Atomwise and just my thoughts on that. Oh, and before we get started, if it's okay with Randy and everyone else, if you have a question, please put it in the chat, but I'm very open to just you saying your question out loud during the talk. That's perfectly fine. I see this hopefully as more interactive. So please let me know if you have any questions on what I talk about. I'd be glad to take some time to go over it. Okay, so why is it important to find new and better medicines? Well, one of the main reasons that I can think of is we have new and emerging diseases. And as humans go into places where we haven't lived before, viruses and bacteria and fungi are coming in, and parasites, and they're crossing into humans from animals and the environment. And so that has led since 1980 to 2013 on this graph to a 2x increase in the number of unique diseases per year and a 6x number of outbreaks per year that has occurred. And again, the big reason for this is because we're pushing into areas we have not lived before. So as our population increases, as we push into those areas, more zoonotic sort of infections are coming in to infect humans. And here are some examples. There are a lot of new emerging diseases, and one in particular is SARS, so severe acute respiratory syndrome. There was actually an outbreak that occurred in 2003. And that outbreak, what occurred in that outbreak was that in less than 100 days, SARS, so SARS-associated coronavirus, The virus that causes SARS spread to over 24 countries, infected over 8,000 people, and eventually killed nearly 800 people. And so that's just incredibly fast. And a lot of that has to do with our ability to travel and how close we are together in cities. But that disease was able to spread very quickly. And a lot of people think that we actually dodged a bullet in 2003. It's unfortunate that the number of people who died, but it could have been much worse, is what they were thinking back then. And of course, now we're actually in a pandemic also caused by a human coronavirus. This coronavirus is a little different, SARS-CoV-2. But since the pandemic has started, we actually have over 5 million people lives lost to this pandemic. And the reason why I include this picture here, this is a picture of an ICU, and these are a picture of Navy nurses caring for someone in ICU. But I really want to share or just remind us that these numbers, 5 million is a number, but there are people that underlie it, right? These are people who have died and whose lives are affected, their families, because of this pandemic and because of this virus. So what are some of the challenges? So it's amazing, and I was talking to Randy and Tack about this before our talk, that we have this vaccine that's highly effective. It's amazing how fast we've gotten it. It's incredible technology. But what are some of the challenges that we could face? So one of the things that I really want to communicate is that, of course, viruses change all the time. And so you'll hear new variants like alpha, beta, gamma, delta that you'll see in this table here, and when they started to become variants of concern. And really, what do we have available in order to fight the virus and those different variants? Well, we have vaccinations, again, this amazing vaccine that we have. But now, more recently, we're starting to have drugs be available in order to fight the vaccine. There are other sorts of treatments, but these are the main two. But I really want to drive home the fact, again, that the virus changes. And even though we may have a vaccine that's highly effective now, down the road, if it changes enough, that vaccine may not be as effective, or the drug may not be as effective as we'd want it to be. So we do constantly have to do research and check and make sure that what we develop is actually fighting the virus as we want it to. So what does it take in order to make a drug in the first place? Well, I can tell you that it's incredibly complex and costly. So on average, it takes 12 years to produce a drug, costs about $2.6 billion, and has a 97% failure rate. I mean, think about other sorts of industries and where they had those sorts of numbers. It's hard to come up with an example, but drug discovery is absolutely one of them. So what I want to do is I want to provide an analogy of what it's like to come up with a drug. So here I have this mountain range, several different mountains here. SARS, H1N1, so influenza, MERS. We have these different diseases that we want to make a treatment for, let's say. But the analogy is that it's very much like going up a mountain. It's just difficult, really incredibly difficult. So let's zero in on what it means to develop a drug for SARS, for example. So you start off with somebody here at the base of the mountain trying to climb up, but they get lost. They don't know exactly where they're going. It's just incredibly difficult. So they end up somewhere where they really want to end up is at the top of the mountain, where that indicates that they've found something to treat SARS, right? But they're not there. Okay, then you have more people come along, right? They're going every which way, but they're not headed up to the summit. They're not headed to the top. Now, what if those people were able to share and talk to each other and say, hey, this worked for me, but this did not? Could that process be more efficient? And I would say, yes, it absolutely could. Because if you had people go before you and share their experiences and everyone's talking to each other, then the process of actually getting to a solution could be much more efficient. And so when I talk about crowdsourced medical innovation, this is something, in a way, a paradigm shift from the way that medicine is traditionally made. So now you have, for example, pharmaceutical companies that develop these medicines, but they're very much proprietary and in secret. Whereas what I'm describing here is an approach where it's open and accessible to everyone, so everyone can share their experiences and we can improve on medicine based on everyone's work. So it's fundamentally different than the way that medicine is traditionally made. So in order to think about how are we going to produce this medicine, I think it's important to understand the scale of where it is that we're operating. So when I say we want to develop a medicine, at what scale am I actually talking about? So we start off here, a dog, approximately, these are all approximate, a meter, and then we go down to the different sizes here, all going down by powers of 10. So for example, hair is 10 to the negative 4 meters. You go down here to red blood cell, bacterium, and here is SARS-CoV-2, the virus that is causing the current pandemic. And look at how small it is compared to us. So that's roughly 10 to 100 million times smaller than us, is this virus that's causing the pandemic. Okay. Now, when I talk about a target, as you're going to hear later in the talk, I'm actually talking about a protein, which is roughly 10 to the 8 meters. And then when I talk about a small molecule acting as a drug or a medicine, that's 10 to the 9. So here's an example of a sucrose molecule, and that's nano. So the prefix for that is nano. So we're talking about the nano world is what I'm going to talk is what I'm describing. Okay, so this is such a small world, like small scale, and we represent these structures using computers. So I wanted to give some examples as to what those structures look like, the different structures and the way they're going to be represented in this seminar and all the others that are to follow. You have 3 basic types. There's going to be the stick, and that's where you see the elements, and we're going to describe this more. But these elements come together and they stick together in these bonds, and that's going to be the stick, or what I like to think of as the skeleton representation. Then around that, when you talk about these atoms and these elements, they're really like these spheres, right? They're really these spheres stuck together. So that's what's known as a van der Waals representation here. And the different colors I'll talk about, those are just the different elements there. But I like to think of that as kind of like the muscle. And then finally, you'll see the surface. So imagine if you put like a blanket around the van der Waals representation, and that's going to be the skin or the surface. Now, when I talk for the rest of this seminar and the ones to follow about intermolecular interactions, these interactions that occur between molecules, they're occurring at the interface of skin. So it's that contact. So if you were another molecule and you were to meet another molecule, let's say in that scale, this is what you would actually see. So you're going to see these different types of representations. And again, I just wanted to point them out here as we prepare to look at that world, more and more at that world. Okay. Okay, so now let's take a closer look at the SARS-CoV-2 virus. Just the virus particle would be the single one by itself. So this is, maybe you've seen these pictures before where you have basically this sphere and you had these spikes that come off of it. This is a 3D printed representation of that. So you have the spikes right here, and that's how this virus enters the cell. And this is going to be my cartoon representation of it. What I really would like you to take away from this is that there are what are known as non-structural proteins. These are like gears inside. Think of them as gears inside of the virus particle. They help the virus to do what it does. The machinery behind it. And then you have structural proteins like the spike. They help the protein to get into cells, as we're going to talk about. And then the third part, which is really important, is the RNA. So this is going to be like the instruction manual of how to make more and more copies of the virus. So those are the 3 main parts of the virus represented by this cartoon. Are there any questions on that at all? Or good? Okay. So this is a movie that I developed kind of representing how, in general, this sort of infection would take place. So you have the virus go through the bloodstream. In extreme cases, it will go through the bloodstream, and it'll deposit the RNA into our cells and hijack the cells. And instead of our cells producing what it needs in order for it to live, it's hijacked, so now it is producing the precursors to the proteins that the virus needs in order for it to make copies of itself. And then there's something very special here. This scissors here that's breaking up this long string of proteins. That's what's known as a protease, and I'm going to talk more about that. But essentially what it does is it takes this long string of protein, cuts them up so that new viruses can be assembled. Those viruses then go crash through the host cell and then are spread throughout the body. But when we think about how we want to make a medicine, what if there was some way that we could gum up the scissors in the first place? So now the scissors cannot cut up the proteins that are needed in order to make new virus. So you've essentially stopped this the replication, what's called the making of copies of this virus, and then that gives your body enough time to produce antibodies to fight off the infection. So you're giving your body the winning edge here. You're giving it time essentially by doing this. And so though you may still get the virus in you, we're preventing the virus from making copies of itself, and that is the key to preventing SARS from then killing people. Okay, so what does this protein look like? When I talk about a protease, when I talk about scissors in this analogy, it very much is like a little machine. It's just incredible. It's like a nano-sized little machine that moves. And I want to really emphasize that. Everything that I talk about here, even though you may see it as a static structure, these things are highly dynamic. They're always moving. They act like little machines. And when we actually look at that machine that I'm using scissors as an analogy for, it has a portion of it that's actually moving as if it's cutting up the polyprotein, what I talked about, that long string of proteins. And so the same idea, if we can think of some way to gum that up and make it so the protein cannot function the way it normally functions, then we can prevent replication. We can prevent this from further harming the patient. Okay, so how do we know what things look like on that scale, on the nano scale? Well, there's actually something called X-ray crystallography. And what it does is it takes this highly mobile protein and it takes a snapshot of it, as you see over here. And we're going to learn more about these representations in color. So I'm going to talk more about that. But the main point that I want to get at is in this scissors, there's the shear part. There's the part that actually does the cutting. And that's what we're going to call the active site. And so I'm going to zoom in on the active site in this video here over on the right. And you'll see that parked inside of that active site is a small molecule. And we're going to talk more about that. But this small molecule you can think of as acting like that gumming up sort of thing. It sits in that pocket, in that active site, and it prevents the protein from doing what it normally does. And that's the secret. That is really what it takes to develop a drug typically. That's not the way that all drugs work, but that's the way that most of them do. Okay, so what sort of challenges might we face there? Well, first let's zoom in on what that molecule looks like. So you see gray, red, white, and blue. Those all correspond to different elements. They're mostly nitrogen, oxygen, carbon, and hydrogen, as we're going to learn. But I want you to think about the small molecule as being a key. And it has a special pattern, just like this molecule has a special pattern. But you can break up that molecule and you can arrange it in just a slightly different way. And it's going to have totally different properties and shape. And that's indicated by this other key here that's going to look different. And these properties are going to look different. Now, that's very important that we understand that we can just rearrange molecules in a small way and make very drastic changes. So what are the possibilities? Well, there's 10 to the 60 possible different combinations that we can have. That's just incredible. There's just that number is just it's huge, right? There's just no other way to put that. And then so when we I'm sorry, let me move beyond this slide. Okay, so another way that I want us to think about it is, again, this lock and this key. So the lock represents the target and the key represents the small molecule that we're trying to fit in just the right way into that lock in order to open it. Now, if we open that lock, we've cured the disease. Think about it that way. And so just like when you put a key into a lock, there's going to be those pins, and the key has to fit in just the right way. And so the takeaway here is that when we're thinking about a drug, the drug has to fit in terms of shape into that active site, but it also has to complement the properties of that active site. Those two things are very important concepts that we need to take away. And again, you see this hydrogen bond donor acceptor. I'm going to talk more about that, but I want you to give you a more broad view of it right now, and we'll get more into that. Okay, so we have the structure of the target, the therapeutic target, what we actually want to go over. But the issue here is we don't know what the key should look like. There are many, many, many possible keys. We just don't know which one is the right one. So really, when we're talking about designing medicine for a particular disease, typically we know what the target looks like. We know what the active site looks like. And it's almost as if we're reverse engineering from that structure, we're reverse engineering what the key should be. And so I'm going to describe in this video here how we've gone about that process before. So I don't show it here, but we actually have a paper mache model. And what I've had my students do is they put together these plastic molecule kits in a particular way. Then they use a user interface to put that molecule and submit it into web services. The web services do the calculations and then give back what I call a report card, a molecular report card. And that report card has a therapeutic grade point average. And this is important because all students, I'm sure, want to get a higher and higher GPA. That's the goal. And so this has allowed me to gamify the approach. So the students will come up with an idea. They get to be creative. They get to try different things out, and there's no wrong answer. Then they submit it to calculations, and then I'll describe in a little bit how the computers go through those calculations. And then they get results in the form of a report card, and that helps them to refine their idea. So there's this constant process of trying to do better. And that allows this gamification of drug discovery, and enables it, opens it up to everybody. And then through that gamification, you can also share your results, and everyone gets to be a part of it. And so behind the scenes, when we submit these to the computers, what is the computer doing? Well, if you think about a key going into a lock, you got to move that key sometimes to make it fit just right. And the computer is doing a similar thing. It's taking that small molecule and it's trying to fit it into the active site in just the right way. And there's a score that's associated with that. We call it an energy. But that score, you want that energy, in our case, to continue to go down. And so that process is what the computer models. And then it gives it a score, and then that's how the students know where and how to refine it, and then they go about that process. And so it's been really nice and wonderful to be able to do this project, because so far we have nearly 2,000 ideas for medicine. They're created by students and counting. We pursued targets such as Zika fever, SARS, the original virus, the original outbreak that occurred, staph infections, cancer, and Alzheimer's disease. So really what I want to communicate with this slide is that there are many different targets out there. And what we really need are people to be creative and come up with ideas, because we don't know what the right idea is yet. We have to reverse engineer that. But this process allows everyone to be a part of that. Okay, so in summary, like, what is it that we're doing? What is it that we're trying to do? So, like, it doesn't have to be a virus. It can be a bacteria or some other infectious agent, or it can even be something in the human body like cancer. But the idea is that there's some sort of protein in there, some sort of very small machine that's going awry that we actually want to stop, right? And so that's what I'm calling a therapeutic target. And there's a very special place on that therapeutic target called an active site. And then what we do is we don't know what the ligand is. That's what the small molecule that's supposed to fit in that active site, just like the key. We don't know what the key is. We're trying to get at that. And so what we do is we bring together the students, computers, and this sharing and crowdsource approach in order to come up with ideas for what that ligand should be. And we get better and better at it because everyone gets to share their ideas, and we have a way of scoring those ideas. And then that ligand, we use a computer to what's called dock, and I'm going to talk more about that, but to dock it with the active site in a model. So we dock it, and then the model tells us how good that docking should be. And then those fits that are good fits are likely to become good medicine as well. And so we can design a ligand that's going to prevent the virus from replicating, prevent it from from doing what it normally does, and then that protects people. So what I'm going to talk about in the next seminars, and we're going to talk a little bit more about biochemistry and the elements of the periodic table, bonds and how molecules are put together, the shapes of molecules, the properties that they have and how they interact, and how they move, because all of these are important ideas that ultimately help us to design medicine. And so that's what I'm going to be talking about in the subsequent seminars. So now I would like everyone to try this out for yourself. You can actually try this process out for yourself, where we're taking a small molecule and we're docking it into a protein. So if you go to Durant Lab, pitt.edu, and there's something called Webina. And Randy, if you're still there, I know Randy put it in this space.
Yep. So now I think you can all hear me. If you go, if you look at the map, there's a table up to the right, and it's probably the second table up there, and you can just go click on it up there. Cool. And I'll go over there right now. There we go, folks. And so you should, hopefully you can all hear me.
All right.
And Ben, you can probably, do you need to broadcast now, or should we all just be in the circle?
I do want to go over a few of the steps, and then a lot of it, you can experiment with, and I invite everyone to explore it. But I do want to kind of go in general, just what the next steps are. There aren't that many.
Okay, so if you want to do a demo, and then folks, if you click on that webinar activity thing, it'll come up in a new tab, and you'll be able to do it after you've seen the demonstration he's going to do.
Right. So when you go to the Durant Lab, what's called Webina, this is doing that sort of computational bringing together of the ligand and the therapeutic target that I was talking about, and trying out different ways of putting in the small molecule. So if you go there, this is going to be what you see for the page, or what you should see. And down here at the lower right, you'll just click on Use Example Files. So you click on that. And then what you're going to do is it's going to populate with these windows here. And you're going to see the small molecule here on the right. And then on the left is going to be the therapeutic target with the small molecule already in it. So you can use your cursor to move these around. They're visualizations, and it gives an idea of where the small molecule is going to be tried out in the active site, and gives an idea of what an active site looks like. You can also play around with these numbers here. This gives the size of the box and the position of the box. And you can just try different things out. And then finally, when you're done and you want to actually run the calculation, you just go down to the lower right again, and you're going to see Start Webina. It should take a couple minutes, depending on your resources. And then you should get this as a final result. So you're going to see this protein. Again, we're going to talk more about it, but this is a surface representation with the sticks, like I told you before, the skeleton and the skin here. And you get to see the results of the computer generated. And you can do that just by clicking on each of these lines, and then it should show you what that different way that it put the molecule in the active site, what that looks like. So again, I invite you to explore it. I think it's really helpful in terms of understanding where we're going. You may not understand every aspect. That's okay. We're going to talk more in depth about it, but I wanted to give everyone kind of a big picture look at what it is that we're doing. And we'll delve into more detail next time.
Cool. So everybody go ahead and give it a try. Let's give it a try for maybe 10 minutes. And hopefully that'll be enough. If you need more, that's okay. Feel free to ask questions as you're going too. I don't think you're able to share your screens under the present, but if you've got a question, you can, we'll figure out how to share screens or something, how multiple people can share screens. I can make you a co-host or something if you want to. And then if you just have some general questions, right? This was our cool overview. It's really, it reminds me of what got me all excited about the project to begin with, right? It was like so cool. And for those of you that are working on the project now, Danny and Tristan, I think that gives you some of those visualization options that we'll be talking about and what we'll be doing. But it'll give you this overall target. So go ahead and play with the activity right now for about about 10 minutes. And then we'll just, we have a few things to do as we finish up, and I'll go over those. But you can just kind of hang out in this space right here. Cool.
My Webina is still running.
Yeah. How long has it been?
Oh, maybe like a minute and a half.
Yeah, it takes about it depends on because it's all running in the browser. So it depends on the resources that might be available.
Oh yeah. Yeah,
Yeah.
I'm sure that Kumo space is taking up some space.
Yep.
Yeah, so let me know if it just does not work for you.
At what point would I be like, this is not working?
I've seen for me the longest is maybe 5 minutes. If it's not,
Oh,
if
okay.
it's not
Okay,
going,
so not like 20 minutes or something.
no, no. Yeah,
Okay.
there's definitely something wrong if it's 20 minutes. Yeah, yeah. All right, let me see if I can. Okay, so you guys should be able to see my screen now, yeah?
I cannot.
Not yet.
Oh, let me try that again.
Oh, I got my output.
Cool. And if you want to,
Okay. Now,
you
yeah,
can.
you should see my screen.
Is
Yep.
your
Okay,
output
perfect.
a
Yeah,
link?
so there's a few things that I want to point out. Again, if you're not at this point, please let me know, and I'd be glad to help out. But all this is just the example files that are showing. And this box, this yellow box that you see here, that's going to be where we tell the computer to sample. So just again, like if you had a key and you're trying to fit it into the lock, you don't want to try it just anywhere. You want to try it just in that lock portion of it. And that's what the yellow box is doing. What you see here in this yellow molecule, that is what we know to be through X-ray crystallography. That's what we know to be the shape and the position of the molecule. Now, what we can do with these simulations is we can compare it to what we know it should be. And we can see how good these simulations are. And again, these are abstract models, so they're not perfect, but it gives us an idea, a benchmark, a way to determine whether we're headed in the right direction. Another thing I want to point out that we're going to talk about later are these kind of like these. The best analogy I like to describe them is imagine you have a car and it's going through a factory and you have those robots coming in and they're doing different things to the car. In biology, these are what are known as residues or side chains, and they're essentially doing the same thing. You have a molecule come in and these side chains it's almost as if they're operating on that molecule and doing something special with it at the active site. And so I wanted to point those out. We'll learn later what these colors mean and what the ribbons mean, but this part here, where you see that yellow molecule surrounding it, that's the active site. And it's a pocket. That's essentially what it is. And it's really that we're going after. That's like the pins in the lock analogy that I used. And then this gives you a 3D representation of the molecule over here on the right. And again, I want to point out how these molecules can take on different shapes. They're highly dynamic. I know we see it as a static picture here, but this would be moving like crazy in the real world. And so that's what the computers try to capture, is that sort of movement and interaction. So I wanted to show you that. And then I'll make mine run. You guys, you can continue doing what you're doing there, and we'll see what we get in terms of results. Anyone having any issues on it or any questions?
No, mine just ran and it took about 2 minutes.
Okay, yeah.
So not too bad.
Yeah, not too bad. And I think with I think I got like try to max out my RAM on this new computer, so it took me a minute.
Right.
So I think mine yeah, I don't.
Right. So another thing you can do, and so we've talked about these visualizations, and we'll talk about this more at another at one of our following ones. But if you go down to the Moleculeze project progress, you'll see where we're going with the project. And Danny and Tristan have programmed the start of our tutorial level. So you can imagine that before you can do this well, you need some knowledge. Now, most of you probably have enough of that knowledge to do some of it, but our goal is to start from no knowledge and bring it on forward. So we've started just with colored balls as being that. And Danny and Tristan, you can now probably see forward to some of the structural representations that will come from this, right? We've got all three of those models. We've got the stick, the muscle, and the skin molecules. And I think with the CSV that you worked on, you could probably figure out a way to do all of those models, right? So so already you're starting to think about that. But if you want to go down and just check it out, you can play our most current version, which is basically just learning how to select different colors right now. Danny, you had, I think, some questions about how do we, while they're learning a skill, how do we prepare them for the next level, right? Like, where
I
do we
guess.
need certain information? Have you put any of that in there yet?
Well, I have it all kind of like laid out in a document of like what I want the tutorial to look like and what I want some of it to look like. I do have like an example of what something might look like in. So I wonder if I can just give this link. If you look.
If you put the link in the chat, Professor
Yes.
Samudio can pull it up.
So if you look at this presentation, I'm so used to Discord. Give me a second. It's on the 7th slide.
Where did you put it?
In the chat.
Okay, there we go.
Current room. Do I need to put it?
No, he found it.
Rooms?
That's a nice logo, by the way. I really like it.
Yeah.
Thanks. Yeah, we talked about making it more accurate, though.
And before you leave, if you would go to the meet and greet area down in the lower right, there's a poll. There's a number of different versions that Danny has made, and we'd like your opinion as to which one you would be more interested in seeing.
Yeah.
But yeah, so that 7th slide. So I'm. My question is, so now that I know more about like the process and the program and everything, kind of my question is more of when you were working with students, you were building them for like the talent search program for Sac State. Is that right? So when you're working with students for this, do they know what all the colors are? Like, do they know that carbon is black and that hydrogen is usually white? What's their level of knowledge when they come in for that sort of thing?
Typically, no, they don't know what those are, and what those colors are. Since you mentioned color, yeah, they don't know what that. I would say that they've heard of elements, they've heard of atoms, but there are different representations. This is one of the most common where you see carbon as black, but they don't necessarily know.
So how big is that learning curve from them coming in and then to them making a model of something and submitting to you? How how long does that take to kind of teach them everything up to that point? Because maybe that's something that I can focus on with you. This game is that kind of thing.
Yeah, no, that's a great question. What I would say when I was working at Sac State with the talent search there with those students, they were in elementary, sort of that elementary school, and it took I would say about 3 days. But remember, I was only maybe meeting with them maybe an hour or two a day, something like that. So in total, it wasn't a whole lot. I think part of the reason why is we had these plastic molecular kits so they could hold it with their hands and they can piece it together like Legos. And so they really enjoy doing that is what I found. And so when I would tell them like this, black is carbon, nitrogen is blue, they can start to put those molecules together right away. In other words, I didn't need to teach them the mechanics behind that. They could do that. And then because of the way that these plastic molecule kits are built, they have different spikes and different holes that fit in just the right way so that they couldn't, how do I say this? Like they would get the bonds correct just by piecing it together like Legos, right? They couldn't get a wrong valency for carbon, for example. They would always get the right one because it only has 4 holes and 4 holes to be able to fit the spikes, for example.
Got it.
So I would imagine if you wanted to emulate that in a virtual environment, you absolutely could. And I've seen examples where they'll have the atom, and I'll have to point you to something called PhET simulations, but they have the atom and it will only allow you to do a certain number of connections to another atom. So even if you don't necessarily understand valence electrons and those sorts of things, just the mechanics behind it help you to see that, oh, there's a limit to how much I could put.
Got it. Okay.
Good question. Definitely really nice.
And do they know when they're putting them together, do they know, oh, I'm putting, 2 carbon atoms and, 1 hydrogen so that it'll do this specific thing? Or are they just kind of just throwing things together and going, how
Yes.
about this?
Yeah, initially, yeah, absolutely. Initially, they are just throwing stuff together, and I actually invite that. I actually really like that because I want them to play and explore, because I actually think that that's the avenue to real learning is when you get to have that opportunity to just play and have fun with it. So I give them some time to be able to do that. But then what I start to do is I start to introduce things like oil and water. Let's just think about oil and water. Okay, now this is the way water looks. This is the way oil looks in terms of molecules. Now you look at the molecule that you're building and you tell me how much of your molecule looks like oil and how much of it looks like water. Because depending on that mix, I can tell you whether it's going to be able to go into cells or not. And cells is where you typically have these therapeutic targets that you're going after. So then they start to get this idea that, maybe it's not a good idea that I string a bunch of nitrogens together because that's not realistic. What these molecules typically look like are a lot of carbons, and sprinkled among those carbons are some oxygen, some nitrogen.
I'm starting to get a better idea of what we should do next with our project.
Right. Yeah. And so that will be, and that's one of the things that we were starting to look at when we were doing the VR version of this is, there's like we've talked, Danny and Tristan, well, let's just plop molecules into here and make them attach together, and that'll be fun, and we'll learn something, and that'll be good. But at some point, we want to be able to build molecules that will fit. And so a whole nother little branch of this is going to be, I'm going to build a molecule and then try to put it into place. Like we're going to build that one on the right-hand side of the web builder, the one that we did. I want some that are pre-built, and then I want to be able to build my own, and then I want to be able to make them move around. So we've got a lot of cool things to do, but I like your idea, Danny, and like this one looks really cool, right? Oh, you just played with carbons. Here's a little bit about it, right? And as you move up, you can add more information, maybe. But this is like a great start one, right? Like, oh, get all the black ones. Click, oh, black means carbon, blah, blah, blah. So I really like this concept of starting at the very beginning and just, you're still playing, but you've got these little pieces, these little spike proteins of information invading your brain, right? They're injecting little chemistry knowledge into your brain. I like that.
Hey, what about making a virtual chemistry kit or the molecule kit, I put a link into the chat channel, because we can even build in templates, because a double bond works differently compared to a single bond. The lengths are different.
Yep.
So there are certain rules about, if you put this particular atom next to this particular atom, the single bond length is going to be this long. Or if you put a double bond, okay, it's going to be a little bit more rigid, because you're basically using two little pieces of plastic, both kind of flexible, in order to make two of the balls connect. But you cannot twist anymore, because you can, but it's harder. It cannot freely rotate anymore. But we can actually build in a lot of those as templates into a game, where people just have to identify and go like, okay, I see that this particular ball has this socket and this socket still open, so I want these two sockets to connect to this double bond thingy, and then the other end of the double bond thingy will connect to those two spots of that particular atom or that particular ball on the other side. So this way, people don't have to think much anymore, because it either snaps or it does not snap. But all the
Exactly.
snapping are basically template based, so that way people can put things together in a much more rapid fashion. But everything is
Yes.
still having a chemistry foundation, because they are basically just a virtual version of the chemistry kit that we can buy off the shelf.
Right, absolutely. No, and I like all of those ideas, and I like the way that Danny has the like just trying out the here's a carbon, identify that first. Like it's this training that you get as you're starting to play this great game, and I can see that as a very valuable educational tool. Another thing is I do feel like there's a lot of ways, there's a lot that can be done with it. So, for example, if I wanted to describe what a dipole moment is across a bond, I can say that in an abstract way and talk about the mathematics, but really when it makes sense to people is when they can see it and they can feel it. And so you with Unity, as I understand it, you have this physics-based sort of engine. And imagine if you can kind of pull molecules in a particular way, and you can see. And again, I'll describe what a dipole moment is next time. But you can see this dipole moment move as you're moving molecules. That's one way, one idea of many ways that this could go.
I wanted to ask, in the presentation, all the videos that you were showing with the molecular models, what software are you using to do that?
So do you mean the visualization of the molecules themselves, or
Yes.
the movie, making the movie?
Uh,
Oh,
the visualizations themselves.
yeah, so that's what I'm going to introduce to everyone next time. I'm actually
Okay.
going to send a link. It's called VMD, Visual Molecular Dynamics. It's free. It's if you're at an educational institution, you can download it. You just need to register. You can do it now if you wanted to, and it's very powerful. There is a bit of a learning curve, but I'll go over some of the basics of it, and we can start looking at molecules. But in addition, one of the reasons why I really like VMD is because you can make OBJ files from it. So you can get something in VMD, make a particular structure, and you can output it as an OBJ file, for example.
And then
What was it called again? You said
VMD, Visual Molecular
okay.
Dynamics.
That's
Yeah.
what I thought. Okay.
right,
Thank you.
and currently, Ben, we're working off of off of your original CSV concept, right? Where we've got X, Y, Z in color, right?
Right. Yeah.
And we've actually just imported that into Unity, and they've attached a sphere to it of a certain diameter based on the color, right? That we've assigned. So does the VMD also provide you the opportunity just to put out a CSV?
Yep. VMD along with the script that I wrote, but the script I put on GitHub, so it's out there. And I do also want to mention that out of the Durant lab, which is where we're getting this Webina, there's also a tool there that makes it super easy to do all of this in Blender. And so I don't know how much the Design Hub uses Blender, but that might be an option.
We do.
Okay. Yeah.
And so and that would be interesting for the programmers to decide whether you want one OBJ to go into there, or do you want an OBJ, or do you want a CSV and develop it on your own? Right? There's I suppose there's interesting pros and cons for each one as you're working away there. So okay, well, we're just about out of time, so I want to close up. First of all, there's no restriction on this link. You can come back in here whenever you want, and either look at the webinar activity or go down below and play the game, and go get a cup of coffee or whatever. You can even meet in this space on your own if you want. So there's no restrictions on coming into this space. And so if you would, please, you saw that we have a very cool logo, but there's versions of it that we're interested in finding out your thoughts. There's colored versions, there's animated versions, there's black and white, and there's simple and some with some other stuff. And Ben, one of the things that we were talking about was, do we need it to look like, 2 on the inner ring, 4 on the outer ring? Does it have to represent something real? Or, like I said, the gear thing where everybody always uses a gear set that actually doesn't work. So, if you think it should be something, your input down on there would be good too.
Well, I already changed it to 2 on the inner and 4 on the outer.
Oh, okay.
So.
Yeah. I mean, if you talk to a hardcore quantum chemist, they'll tell you that none of those representations are physically correct,
right.
And they're right in that, because the reason why is because we're limiting it to a 2-dimensional space when it should actually be 3. But everyone uses that representation, so it's fine. But yes, you're right. There are 2 inner, those core, well, depending on what element you're talking about, but 2 inner electrons for the 2s orbital, or 1s orbital. Yeah, that's right. And then it goes on from there. That's right. So no, absolutely fine in doing that. No problem.
Yeah. So that's
and also the benzene molecule looks like an O as well. It is a little ring.
Right.
Yeah, and so when you see that hexagon and you see the circle inside of it, that's shorthand for these double bonds that alternate.
Right.
You have double bond here, single, double, and I'll talk more about that next time. But when you see that circle, it's just shorthand for that.
And I think the benzene molecule is also a very interesting example of how the double bond is limiting how the rest of the molecule can move or not move in this case, and that's why the benzene molecule has to be a circle on a plane. There's no other way for it to go.
It's flat. It's flat. Yeah, definitely.
Yep. But having a physical model, makes it kind of really, if you actually have hands on, and actually try to plug those things in, you will find that the last few things that you want to plug in is like, it's hard to plug, especially the double bond. And I think that really is something that cannot easily be replaced by a virtual environment, because you cannot feel the tension of the little plastic sticks that you need to plug in.
Actually, with good, Tristan, you can tell us, but with good haptic feedback, you could, right?
Yeah, that's that's the next thing that I want to bring up is, how much of the Unity stuff that we have done so far, how much of that can be used using a VR headset? And the second question is, can a modern VR headset like the Quest 2, can it utilize a Unity program that is using WebGL, through a web browser?
Right.
So I think those are questions I want to, throw out.
So if I could just, I'm going to have to show you at some point. I talked about paper mache. I didn't show it in this seminar, but what we did is we made paper mache of an active site, and we made augmented reality so you could actually see the results of the computer overlaid on the paper mache to scale. So it's a model that's at scale. for the active site. So I know that it's a possibility. I used A-Frame. It wasn't it wasn't that difficult to implement. So I would imagine, I haven't tried something like Oculus, but I would imagine that you could do both AR and VR in a pretty straightforward manner, especially if you're using Unity.
Right. And so we do have the capabilities, and we were working toward VR, but one of the things that we found with our shutdown and the COVID and the pandemic was that a huge number of students that we could reach don't have access to those.
Yep.
And so we've purposefully at this point pulled back to WebGL versions of a Unity publish that can go on basically a Chromebook for that very reason, for the accessibility. And then
Right.
Tristan can maybe speak to how hard or easy it is to reprogram to a VR environment, if we want to do that. But first, I'm going to have to leave soon. I want to do two things. So a couple of you who are here are not on the project right now, and we had a few people that expressed interest but could not be here for this one. So while the recording is still going, I want to mention that if you're interested in working with us on this project next semester, even if you're already in the Design Hub, please send me an email, and I'll send you the information on how to become an intern at the ARC Design Hub, or if you're already an intern and you go, I like what I did this last semester, but I want to do this now. Let me know. It's schustr at arc.losrios.edu, and you've all got it because you're here. So there we go. So at this point, I'm going to go ahead and turn off the recording.